So this lecture, we will go to talk about non-spore forming gram-positive bacilli. So the gram-positive cocci is over, okay? Strep, staph over. Now we think about gram-positive bacilli. For gram-positive bacilli, we classify bacteria into two main groups: can produce endospore or not produce endospore, okay? The one that produce endospore we learn tomorrow. So today we learn non-spore forming gram-positive bacilli. In this group contains so many bacteria. We separate them by morphology, by shape of bacteria. The first group is bacteria that have regular shape. Well, when you think about bacilli, just beautiful bacilli. And stain property like gram stain. When you stain gram to bacilli in this group, you see the purple, bluish purple, smooth. Okay, this is regular thing. We talk about Listeria, bacteria in genus Listeria and Erysipelotrix. Another group is irregular shape. The shape is kind of bacilli, but it's not regular bacilli. They look weird. Some of them look like a club, like a ball. So one pole is bigger than another pole. Or some of them look very long bacilli and branching bacilli. That is filamentous branching cells, or some process different type of cell wall like acid-fast bacilli. That's the thing. You learn all of this in this lecture. So we cannot go to very detailed, so I jump from one bacteria. I try to emphasize just only important point, okay? Let's start from Listeria. In Listeria, well, just only one bacteria that I like to mention is Listeria monocytogenes. I think you probably heard about this bacteria before, right? Listeria mono is a food poisoning. They cause food poisoning. The structure of Listeria mono is just regular coccobacilli, not long, okay, not long. But, well, some textbook probably say about short rod, okay? These bacteria, when they grow in 28 degrees Celsius, they process Perithricus flagella. 1 to 4 Perithricus flagella is mean like they can move. But at 37 degrees Celsius, once they infected to human, they cannot synthesize flagella, so they non-motile in human. But outside of human, they can move. Listeria is aerobic, and also some strain of Listeria can be microallophilic. They need oxygen, but not much. Okay, Listeria cannot produce capsule. Okay, no capsule, sometime moved and sometime not moved. But if they move, they not move regularly like the other. They use flagella propel around, and cells kind of tumbling movement, end to end like this. This bacteria resists cold, resist to heat, resist salt, extreme pH, and resist by salt. They resist by salt, that means they can infect human by GI tract. Okay, they resist pH. That means they can pass through your gut, your stomach, low pH. Okay. culture of Listeria mono, you probably need to enrich bacteria before you culture. When you get specimen, put the specimen at 4 degrees Celsius for a while. This bacteria is psychophile. They love cold temperature. Well, this is very dangerous. Bacteria love contaminate in food. When you get food in the refrigerator, you think it's safe. Well, regularly safe from the other bacteria, but not safe from Listeria mono, because 4 degrees Celsius is optimum temperature for them to grow. So they grow happily in the refrigerator. And once you put it up like and eat those food, the contaminated food, so you get food poisoning. The media for growing Listeria mono is blood agar, but the base media need to be Muller-Hinton agar. Still remember this name, Muller-Hinton agar? Kind of familiar? This agar used for antibiotic susceptibility test. Okay. Colony of Listeria mono is small, white gray color. Okay. It can hemolyze, but the hemolysis zone is so narrow, almost touch the edge of colony. So sometimes, if you not look properly, you probably see no hemolysis. Okay. Normally, L mono present in water, in sewage, and also in soil. Okay. Once they present in soil, they get infected with vegetable. Okay, and meat, in pig, in cow, in whatever. Once they contaminated in cow skin, the farmer get milk from cow, so L mono contaminate in milk, L mono contaminate in vegetable. If you buy vegetable from the market and not clean properly, L mono will spread. Okay, and well, basically, when you buy meat and vegetable and put in the refrigerator, you not mix meat and vegetable together, because L mono from vegetable will cross contamination in meat, and then that is more serious. They love to contaminate food with high protein and high lipid. Okay, this serious. People who get infected, L mono was outbreak in the US long time ago by together with mayonnaise and coleslaw, because you eat raw vegetable, right? You not cook vegetable. That's high potential to contaminate with L mono, and you keep the coleslaw and mayonnaise in refrigerator. That's the point. L monocytogenes cause disease called listeriosis. Oh, sorry, listeriosis toxin. Okay, the serious from pregnant women, from newborn, immunocompromised. Okay, that's the point. This is differ from the other bacteria you learn. This one is intracellular infection. One L mono infect to human, they not stay outside of the cell. They go inside host cell and survive inside the host cells. Okay, this is mechanism that make L monocytogenes escape immune response, because immune response will not penetrate into your host cells. Okay, first thing, L monocytogenes will attach to the cells on GI tract. Okay, they come from contamination, attacked by the protein on the surface called internalin. Internalin A. This is internalin A attached to cadherin. Cadherin is host cells. Once interleukin binds to E-cadherin, this signal forces the host cells phagocytose bacteria inside the cell. So bacteria command host cells to phagocytose them. Once bacteria go inside the cells, they kind of stay in the vacuole, the vesicle, right? The food vacuole, actually. The general process of cell is this food vacuole or vesicle will fill with lysosome. Lysosome contains so many hydrolytic enzymes that can digest bacteria. Okay, that is normal process. But bacteria also produce listeriolysin O. That is a toxin that digests the membrane of vesicle. Once vesicle membrane lies inside cytoplasm, bacteria escape from vesicle into cytoplasm before the vesicle fills with lysosome. Then bacteria survive, okay? But as I mentioned earlier, at 37 degrees Celsius, bacteria cannot move. Once bacteria escape from vesicle, they have some protein that depolymerize actin, make actin in the host cells, chop down actin, and pull actin unit molecule around bacteria and repolymerize actin around bacterial cells. Once actin polymerized around bacteria use actin for movement, look like a comet tail. So bacteria move inside cytoplasm by the comet cell. Bacteria can move from one cell to another cells, nearby cells by penetrating and form double layer membrane. Double layer membrane go inside another cells. With this technique, bacteria never meet immune response, immune cells, okay? And bacteria likes double layer membrane with listeriolysin O and go another side. Bacteria can pass through the epithelium to deeper area of the intestine. And also bacteria can move inside if the bacteria pass through the layer of epithelium. Macrophage can eat bacteria, okay? Bacteria resist macrophage vesicle by listeriolysin O and survive within macrophage. Now, macrophage need to move through the blood circulation, and that means macrophage bring bacteria to everywhere in your body. That's the smartness of L monocytogenes. Okay. So incubation period can be long up to a month. Well, when you get contaminated food today, maybe next month you get symptom. It's not very quick like E. coli. When you eat today, tomorrow you get symptom, or maybe this evening you get symptom. That's enterobac. So you probably get fever, muscle pain, nausea, diarrhea. In pregnant women, if pregnant women get infected, she probably get symptom like flu, like but the symptom is milder than flu. High fever, shake, muscle pain, something like that, or premature delivery. Or bacteria can go to infect newborn. That's even more serious. If they spread to the nervous system, you probably get encephalitis. That means headache, sore headache, and stiff neck. Stiff neck is mean like your neck cannot, kind of stiff, you cannot move, or bending head hardly, not easy. Confused, this is kind of psychic, and lost balance, you cannot stand balancedly. Or bacteria can cause pneumonia, meningitis, encephalitis, sepsis, or even die. That's the symptom caused by L monocytogenes. Okay. So treatment of L monocytogenes, we use ampicillin. Well, people who get infected, even not so many people, but infected with L mono cannot go to see doctor and get the medicine, go home. You probably need to stay in hospital. That's serious, because I think the treatment is intravenous. You probably put the IV. Okay. Prevention of L mono. Just avoid the food that can eat directly without cook. For example, sausage, hot dog. So hot dog you can eat cold. If you buy hot dog from supermarket, if they get contamination, well, supermarket probably get better brand, is kind of better. But if you buy from unknown brand, in hot dog they probably have some water inside. In the water, it might contain L mono more than inside hot dog. So be careful. And high protein, high lipid, like cheese. Cheese is very important source for L mono contamination. Coleslaw. Try avoid eat raw vegetable. But if you're not so well, clean vegetable properly, that's help a lot. That's the thing. So this is summary of the L monocytogenes. Harm in immunocompromised, get high risk in pregnant, infant, elderly. Okay. The second one, erysipelotrix, luciopathy. So this bacteria is gram-positive, non-spore forming, gram-positive bacilli. So you probably see the long bacilli. Colony is transparent. You probably hard to see a colony on blood agar. Okay, and sometimes in TSI, well, you learn TSI a lot next week. TSI is test for sugar use of bacteria. Mostly tests used for gram-negative bacteria, but erysipelotrix can produce hydrogen sulfide that you probably see the black color inside the TSI. This is the general characteristic of erysipelotrix. Erysipelotrix will concern in veterinary medicine. They infect pig mostly. Well, they can cause infection in so many animals, but mostly in pig. In pig, they cause the red rash on pig, and they cause, well, we call erysipelas on pig. But if they infect human, we call erysipeloid. Okay, so they probably get infected and cause skin inflammation. Once skin inflammation, you see skin swollen, and inside the swollen has fluid inside, painful. Okay, red on skin like this. Okay, treatment still, this is gram-positive. Penicillin is a drug of choice. If patient who allergic to penicillin use erythromycin. Okay. Then we move to the second group, non-acid fast bacilli, but irregular shape. Okay, irregular shape of non-acid fast. Well, they have tons of bacteria, but we probably think about Corynebacterium and Propionibacterium, two bacteria. start from Corynebacterium diphtheriae. This bacteria cause disease, well, variety of disease, but main disease that this bacteria cause is diphtheria. Diphtheria in Thai we call โรคคอตีบ So the cells look like a club shape. Some part of the cells like bubbling. Some area of the cells very narrow. At one or both pole of the cells kind of swirling as bulk. And arrangement of cells is joint. Two cells join each other by one pole, generate the pattern of V or L shape, this kind of V shape. Okay, if you see the cells touching in one side, cause the V and L something, or in some textbook we call Chinese letter pattern. This bacteria is aerobic. They infect throat, so then they need oxygen a lot. Non-acid fast, cannot move. To grow bacteria, you need chocolate agar. Chocolate agar is pretty much similar to blood agar. When you prepare the blood agar base, you need to waiting until the blood agar base cool down, maybe 50 degrees Celsius, something like that, and then you just add red blood cell inside. But if you add red blood cell, since the media is still hot, like 80 degrees Celsius, 85 degrees Celsius, all red blood cell will lyse. And the color of the red blood cell will turn brown. It looks pretty much like the same color of chocolate that we call chocolate agar. But in this chocolate agar, you probably need to add telluride. So we call telluride chocolate agar is media for diphtheria. Slow growth. Colony is black because they reduce potassium cellulite and produce metallic color. And colony actually is not round. It's show up in dome shape. Okay. Disease by Corynebacterium diphtheriae cause diphtheria. Okay. Because bacteria localize in the throat near tonsil, and then when bacteria grow, they make so sorry, internet has some problem. Oops. Okay. So you hear me, right? Can I continue? Okay. So disease spread from one to one by person to person contact. Well, if one get disease, you probably get infection in the upper respiratory tract, And when you talk or you coughing, bacteria, you generate small droplet of your saliva, whatever. Bacteria stay inside saliva and transmission from person to person. Okay. Bacteria probably spread even you stay different from for about 6 feet. So even larger than COVID-19, right? So be careful on that. Disease cause serious in children, school children, or very baby. Mm-hmm. The pathogenesis of Clostridium, Corynebacterium diphtheriae, they process so many weapons. The strain of bacteria that can cause disease, that be a bit special. They produce toxin, okay? Toxin is the key virulent factor of this bacteria, but toxin is not generated by bacteria itself. There are some viruses that attack bacteria called bacteriophage, right? Corynebacterium diphtheriae also has their own bacteriophage. The phage that infect C. diff contain toxin gene. Once they infect to the bacteria, the phage, the virus chromosome will integrate to chromosome of bacteria, okay? And the toxin gene that stay in the virus can working when they combine chromosome of virus and bacteria together. We call prophage, okay? And then those strain of bacteria that get infected has ability to attack you better than the other. Bacterial infection on throat make the cells, epithelial cell on the throat die by necrosis. And bacteria kind of produce the white part on the throat we call pseudomembranous. Okay. And some in young baby, the pseudomembranous may be big, and then once it bigger and bigger, swollen, the cavity that you need to breathe in and out is blocked. So children cannot breathe. That is very harmful. Okay. So bacteria process toxigenic by produce exotoxin and endotoxin. Well, the toxin of Corynebacterium diphtheriae is AB type exotoxin. You learn, well, toxin can be so many types. For example, exotoxin of strep staph, that is toxic shock syndrome toxin. That's type of toxin. This is AB type exotoxin. You learn a lot more later about AB type. The AB type is toxin made from two big subunits, A and B. Okay. In this case, part B toxin will bind to receptor of the host. The B side bind to receptor. Once they bind together, they split part A and let part A go inside the cells. So A move down to the cells and interfere with protein synthesis. That's diphtheria toxin, the AB type. This can be found in the other bacteria such as Vibrio chlorella and a lot more. You're gonna learn. This is very important. And the toxin gene called tox gene present in lysogenic bacteriophage beta phage. This is the name of virus. Okay. Prevention. We have a vaccine because bacteria itself is not harm you seriously, but toxin is very important. We prepare weak toxin we call toxoid. Toxoid is less potent toxin of diphtheria. Okay, that's mean we can vaccination with diphtheria toxoid, but this vaccine come in the set of combination of three diseases together. One is diphtheria, the second one is tetanus toxoid, and pertussis toxoid. So three toxins make it less potent. We call DTP vaccine. You probably need to know this vaccine because when you learn the other bacteria like tetanus or pertussis, we probably need to talk about DTP again. Okay, DTP vaccine injected to very baby 2 to 4 months and boost again about 3 to 5 years in the kindergarten. Okay, erythromycin is the drug of choice in this case. Okay. The second one is Propionibacterium acne. Sounds acne. What do you think about? It is pimple, right? The causative agent of pimple, same gram-positive bacilli arranged in Chinese letter. This letter, I mean, one side of the cell attached to another side of the cells. Some English textbook, they probably say the arrowhead pattern. Some is the V shape, L shape, whatever. The same thing. Okay. This one is usually non-pathogenic bacteria and commensal on skin. They live and get some, well, Propionibacterium acne kind of first line defense of your body also. They're part of the innate immune system. But if they are overpopulation or they live in the wrong places, they might cause disease to you. Okay, and mostly skin infection or GI tract. Super slow growth. If you want to culture in media, you probably incubate for 6 days at least. You see bacteria, and they don't love oxygen much. They can aerotolerant, but they actually this is not an aerobic, should be microallophilic. They love very few oxygen. Okay. And by the name Propionibacterium, they produce propionic acid a lot. The other bacteria cannot. Some strain of propionic acid bacteria used for produce cheese, but some strain are pathogen. Okay. The causative agent is chronic skin disease by block the hair follicle. Hair follicle is mean รูขุมขน and sebaceous gland. So normally this is hair follicle and sebaceous gland is say nearby the hair follicle. Bacteria basically stay on surface, can localize in the hair follicle. When the bacteria overgrowth, they block the hair follicle and cause pimple. Actually, it's inflammation underneath the skin. That is the problem of teenager a lot. That's acne vulgaris disease from P. acne. Then we talk about non-acid fast already, not just much thing. Then we move to acid fast bacilli. The acid fast bacilli, we concern about the genus Mycobacterium. Actually, you learn 10 minutes of Mycobacterium. In general class of medical microbiology, you probably learn 5 hours about Mycobacterium because it's so important. the research on bacteriology, the most advanced research of bacteriology is point to Mycobacterium tuberculosis. So we divide mycobacteria into 3 different groups. One is obligate pathogen, so they harm human. They're not living as free living, but they cause pathogen. The key one is Mycobacterium tuberculosis, Mycobacterium bovis, and Mycobacterium africanum. So 3 of these, they share closely related genetic by any technique of the genotyping, try to type or classify bacteria. We cannot separate 3 of these from each other. But 3 of these show up kind of slightly different symptoms. So we not allow you get infect human and then you know, oh, this is M. bovis, right? So we call the complex of Mycobacterium tuberculosis, Mycobacterium bovis, and africanum as Mycobacterium tuberculosis complex or MTB complex, because 3 of these are hard to separate each other. Another one I didn't mention on slides is Mycobacterium leprae. Leprae is very small bacteria, slow growth, causative agent of the leprosy. Leprosy nowadays, we are less and less number of patients we got leprosy. Leprosy is โรคเรื้อน So Mycobacterium tuberculosis cause tuberculosis, that is one role. So we point this. Another group is opportunistic pathogen. This is important for immunocompromised patients. We get MOTT, the mycobacteria, other than tuberculosis, we got this name. The important one is Mycobacterium avium. By name, this is causative agent of tuberculosis in birds, but they also cause infection in human. Mycobacterium kansasii, okay? Some bacteria in this group that not cause disease is Mycobacterium smegmatis, okay? That's point. Let's start from MTB. MTB is slow growth bacteria, non-spore forming, aerobic, because they infect lungs, so then bacteria need a lot of oxygen. The color is thin and slight rod. We not use gram stain for MTB. Remember in the last week, we use acid fast staining, so then bacteria show up with red color on the blue background, okay? They cannot move. Basically, they not grow on blood agar. They need spatial media called Lowenstein-Jensen media or LJ media. This is a media for MTB. The color is green, okay? And colony is look like this. Well, colony of MTB is differ from the other bacteria. If you experience working on bacteria in the lab, you use sterile loop, touch colony, and then bacteria will stick to the loop up. But bacteria in this group, the colony is very hard and you cannot spread bacteria easily. They clump to be a very stick or rigid, clumpy, something like that. Cell wall rich of lipid because this AFB cells, okay? They infected into lung. The mechanism of infection is very immunogenic. So I avoid to go to the detail because they spend an hour to explain mechanisms. Interesting point is bacteria infected from person to person. Okay. Bacteria, tuberculosis of human and in cow. Cow also get tuberculosis by Mycobacterium bovis, this one. Bovis and tuberculosis and Africanum. Nowadays, we know this is the same species. When they get infected, or people who working in farm or working with infected cow might get infection from cow, or tuberculosis get infection from one person to another person. We cannot, there are some research trying to identify bacteria in the environment, but they not find any bacteria in environment. So bacteria has spread from one to one, and infectious dose of MTB is very low. Just one cell, you can get infection. So bacteria go together with the small droplet of saliva, and then another one inhale the droplet inside, and the droplet go to the respiratory. Okay, bacteria go to localize in the alveoli, and immune cell come to digest bacteria. Okay, bacteria successfully living inside. This is another bacteria that survive inside the cells, called intracellular mechanism. Bacteria go inside macrophage and silence of the macrophage. So macrophage not destroy bacteria, but let bacteria kind of sleep. We call latent infection. We get infected with TB, but TB not harm you at all because they sleep together with the immune until you get elder, maybe larger than 65 years old. If you get complication and have underlying disease, have high blood pressure, diabetes, TB may come back, or you get cancer. In the US, when you read textbook, this is the information you probably need to know. In textbook, we thought that we saved from TB for a long time because the case are dropped every year until HIV come. When HIV coming, they have a lot of opportunistic infection, the disease that live in with us, but since we have good immune response, disease cannot do anything. In the US, the top opportunistic infection of HIV patient is Cryptosporidium. It is protozoa. You learn in Unit 5. But in Thailand, the top opportunistic infection of HIV is MTB. So this is different. But if you read from textbook, you probably get the epidemiology in the US. Okay. Well, TB get infected in human. We have a survey in Thai population, and we expect to see TB patient has age around 35 up, but finally we get the patient who get infection with TB around 25 to 35, that is working people. So most of them are HIV positive. That's HIV let us learn TB never go anywhere, but silent in us. So epidemiology is TB spread throughout the world, okay? And we lost a lot with TB. But the sadness thing is a country that get serious problem with TB are poor country. If you see the red color is high risk of a rate of TB is quite high. That's mostly in Africa. and poor country. Okay, also in Cambodia, something. The gray is also top, but not just the red area, including Thailand. Okay, but red in the US and UK are lower than in Asia, so that's important. Well, we test the TB in the lab by performing tuberculin skin test, injected TB antigen, tuberculoprotein, inject that, and we're waiting for 2 or 3 days. This one you get immune reaction, kind of delay type hypersensitivity, and we measure from this side, this age to another age of the swelling should larger than 15 millimeter, 1.5 centimeter. So then we call positive. Positive tuberculin skin test can be you are infected by TB now, or you get immune response because you recover already, or you get vaccine. So it's hard to say which one. Okay. This thing is also important. You probably need to know based on the research. You kind of scientist. The first line drug of TB, all patients need to take 4 drugs together: isoniazid, ethambutol, rifampin, and pyrazinamide. 4 drugs together for 3 months. But after 3 months, patients need to follow isoniazid and rifampin in another 3 months. So then patient need to take medicine for 6 months. Second line drug is the drug that less resistant, okay? But we put in second line because sometimes side effect is larger than the first line, or the effective is less than the first line. First line is more preferred, okay? You probably need to know the term multi-drug resistant tuberculosis, or MDR-TB. MDR-TB is any tuberculosis that resists to isoniazid and rifampin. We call MDR-TB. So, and XDR-TB is extensively drug-resistant tuberculosis, or extreme drug-resistant tuberculosis. That means MDR-TB that resistant to one of second-line drug. Then we call XDR, because MDR-TB is still used second line to kill, but extreme XDR-TB is very hard. You need to select some drug carefully. Prevention, we use BCG vaccine. BCG is strain of Mycobacterium bovis. It is live vaccine because bovis cause milder infection. We use BCG in children and let children develop immune response. Okay. So now we move to the last group. Last group is small, not so important, but you probably need to know actinomycete. Okay. Actually, actinomycete is very good bacteria, live in soil. When you incubate, actinomycete probably produce chemical that kill the other bacteria. So we get antibiotic a lot from actinomycete and streptomycete species, but well, some actinomycetes can cause disease also. The common disease comes from actinomycetes Israeli, the first isolate in Israel. That's why they name like that. they're non-motile, non-spore forming. This is serious. When you read textbook, actinomyces produce spore, but not endospore. Okay, the other type of spore, not endospore. Spore in this term, I mean endospore. Actinomyces is aerobic. Okay, and this is major characteristic of actinomyces and nocardia. They smell musty, like the earth smell when the rain drop and rain finish. You smell the soil, something like that. Actually, it's not the soil smell, the muddy smell. Actually, it actinomyces smell because they are rich in soil. Okay, actinomyces live in mouth a lot and genital tract, in the GI tract. So people who have poor oral hygiene, something like that, a trauma patient or dental surgery might get infected. That's the thing. Not highly virulent, they mostly opportunistic. That's the thing you probably need to know. Nocardia. Nocardia is another group of acid-fast bacteria, but they branching. If you see the cells quite long and branch a lot. Nocardia has so many species, but most of them are that cause problem are asteroides and asteroides. Staining of nocardia is not acid fast, but staining of nocardia use modify acid fast. Normally acid fast you watch by the hydrochloric acid. But for nocardia, we change hydrochloric acid to be 1% sulfuric acid. That's the modified acid fast staining. Obligate error, they cannot live in an aerobic condition. Okay. Nocardia causes nocardiosis. Nocardiosis is skin infection that damage a lot of skin, can cause cutaneous nocardiosis that attack skin, destroy cellulites, and cause a lot of abscess. And also pulmonary nocardiosis cause infection in immunocompromised and often progress in HIV patient. Well, kind of symptom, they kind of more or less similar like tuberculosis. Tuberculosis nocardia can cause with immunocompromised, both of them can cause lung abscess เป็นฝีในปอด lung abscess. So in summary, so Mycobacterium, Actinobacteria, nocardia differ in oxygen consumption. Mycobac and nocardia are obligate aero. Actinomyces can okay with anaerobic in some condition. But acid fast, Mycobac is acid fast. Actinomyces can use with general gram. Nocardia show up with partially acid fast. But some strain of Mycobacteria, Actinomyces, and nocardia, if you're trying to stain gram, remember last time I said most of the gram cannot penetrate inside that and probably show up with goat cell, but sometimes some strain can stain with gram if you try to stain gram, but the stain is not smooth in the whole bacilli. They look like some area stain, some area not staining, some area stain, not stain. If you observe under microscope, you might see like a beaded bacilli because some area stain, some area not stain, something like that. Okay, so in summary, today you learn Streptococcus and non-spore formation bacilli. It's quite a lot, right? So that's for today. Tomorrow you go on with spore formation bacilli. And the gram positive is over, and then you touch gram negative cocci. And since gram negative cocci is not much, so I will put some group of gram negative coccobacilli, not cocci, not bacilli, together with tomorrow. Okay. Any question?